We go on our second part of lecture devoted to the problems of anatomic and physiological peculiarities and objective and additive method of diagnostics of hepatobiliary system. And as we have stopped on third biochemical syndrome of hepatocellular insufficiency, now we are going to discuss fourth biochemical syndrome, and that is inflammatory syndrome. Usually it is typical for autoaggression. What is autoaggression? When due to some pathological circumstances the organism starts to produce antibodies which are directed into fighting not some alien structures which should be really killed, but the host cells. And if we're talking about, for example, autoimmune hepatitis, we mean formation of autoantibodies with the autoaggression against hepatocytes. This pathological process is called autoimmune hepatitis, and it's traditionally associated with fourth biochemical syndrome, which is called inflammatory syndrome. What are biochemical markers of this syndrome which will arise in the bloodstream and which will be increased, which will be raised in biochemical blood analysis? First of all, that is increased amount of proteins, hyperproteinemia. Why hyperproteinemia? Because those antibodies which have autoaggression, they are proteins. That's why the amount is increased, and total protein, of course, is increased in serum blood. Then hypergammaglobulinemia, increased amount gamma globulins, increased amount of immunoglobulin G in the serum blood, increased T-mole test, significantly increased, great amount of autoantibodies, different autoantibodies, increased non-specific markers of inflammation such as erythrocyte sedimentation rate, C-reactive protein. So you will see all these markers or many of them to be increased in case of inflammatory syndrome typical for autoimmune hepatitis. If we're talking about next method, like biochemical stool analysis, which is also called coprogram, as I've told you earlier. So here you can find out the presence of steatorrhea, which is the presence of undigested participles in the feces, amylaria. Biochemical bile analysis is not used widely in everyday practice. Why? Because you need to carry out duodenal probing to take a sample of bile to send it for analysis, and the process of collecting the bile in general is quite stressful for a child and problematic. Very often the child is reacting with spasm of sphincter of Oddi. It's like reaction typical for a stressful situation, and it's problematic to receive any portion of bile at all. Then fractional duodenal probing is not widely used in everyday practice due to the same reasons which I have explained for biochemical bile analysis. An investigation of viral hepatitis markers is a very important method, of course, of laboratory diagnostics, and actually that is first-line diagnostics when we start to differentiate liver pathology in general. Instrumental investigations include ultrasound investigation of abdominal cavity organs and ultrasound of the gallbladder with holagogic breakfast. What is holagogic breakfast? That is when we give the child something which stimulates contractions of the gallbladder and measure the square of the gallbladder before this and 1 hour after, and compare the sizes, the square of the gallbladder. If it had been decreased for less than 50% from its primary sizes, so we may consider a hypokinesia of the gallbladder to be verified. So it's one of the available methods to diagnose hypokinesia or undercontraction or hypotonic changes of the gallbladder. For example, if to compare this method with cholecystography, which you will be able to see a little bit later on pictures, on slides, it's more easy for everyday practice, and that's why more widely used in general. Though it's not approved in many countries worldwide and is predominantly used in post-Soviet countries. Dynamic hepatoscintigraphy, it's used predominantly in specialized diagnostic centers and is not also used in everyday practice, but oral cholecystography may help us to follow the contractions of the gallbladder, to visualize the stones, the shape, the form, the different anomalies of the structure of the gallbladder, and somehow even have biliary ducts. Endoscopic retrograde cholangiopancreatography is used for diagnosing some liver problems with very hard in general differential diagnostics. And it can be really effectively used for differential diagnosis of, for example, such diseases as primary biliary cirrhosis, primary sclerosing and cholangitis, and is used with this aim. Sphincter Odi manometry used predominantly in elder children and in adults in general, and this is the method which helps us to assess the pressure in the part of sphincter of Oddi. And liver biopsy is a method of final verification of the diagnosis of some liver disease which can't be anyhow diagnosed with any other available methods of diagnostics. So actually it's used for final confirmation of the diagnosis. Also, I would like to mention about such a method which is called elastography. Fibroelastography, that is the method of detection of the degree of solidness of the liver's parenchyma. You know that due to some chronic inflammatory process in the liver, the parenchyma, normal structure of parenchyma, becomes to be replaced with, first of all, fibrotic tissue, and gradually, if not diagnosed timely and if not treated properly and timely, it's progressing up to cirrhosis. So if we are talking about fibrosis, it's a reversible condition. If we are talking about cirrhosis, it's not reversible condition, which can't be with the help of any sort of treatment to have reversible development, unfortunately. So the method of liver elastography helps us to assess the degree of solidness of the parenchyma of the liver. Either there is fibrosis or there is cirrhosis, in order to plan the following medicamental tactics more effectively. That is ultrasound diagnostics, which help us to detect the stone inside the gallbladder. This is the gallbladder with perifocal inflammation. Perifocal inflammation, this perifocal shade, testifies to the presence of inflammation of gallbladder wall, which is called cholecystitis. And this is a device which is used for carrying out of tomography of the liver. No, not just of the liver, but liver may be also diagnosed with the help of this method. And that is hepatoscintigraphy, which shows us accumulation of radioisotopes in different parts of it, parenchyma, and its movement, the evacuation, how it's moving from one part to another. And this is cholecystography, and here there is demonstrated how we can follow the degree of contraction of the gallbladder. So if we don't have any changes in sizes of the gallbladder, it testifies to that it's hypotonic. And if it's too significantly contracted, for more than 2/3 of primary sizes, we may say about hypertonia, too intensive contraction of the gallbladder. Sphincter Odi manometry. This picture illustrates its principle. So you introduce the tube with the device which fixes the pressure at the end of it in sphincter of Odi, and then to the terminal part of common bile duct, and you fix the pressure in terminal common bile duct, in the place of sphincter of Odi, and actually in duodenum, and create the graphic of it. Normally it should look like here, so the pressure in the part of sphincter of Oddi, of course, should be significantly higher than it is in the common bile duct or duodenum. But it shouldn't be too much, that which testifies, which is remarkable for sphincter of Oddi spasm, and it shouldn't be the same as in common bile duct and duodenum, which is typical for hypertonia of sphincter of Oddi. If we're talking about semiotics of hepatobiliary system damages, here we have basically three groups of disorders. Two of them are already familiar for you, and we have discussed it for any other diseases of the gut, and that's functional disorders and inflammatory diseases. If we're talking about functional disorders, the most common in childhood are dysfunctions, dyskinesias of the gallbladder and sphincter of Oddi. They may be of two types. Those ones which are caused by decreased tonus and those ones which are accompanied by increased tonus. If we're talking about flabby, lazy contraction, hypertonia of the wall of the gallbladder, it's called hypokinetic gallbladder dysfunction. And if it's contracting vice versa more intensively, we are talking about hyperkinetic gallbladder dysfunction. And the same principle is active about sphincter of Oddi. If it's too tightly compressed due to increased tonus, we are talking about spasm of sphincter of Oddi, which is also called hypertonia of sphincter of Oddi. And if it's flabby, hypotonic, and doesn't hold the pressure like it should be, it is called hypotonic sphincter of Oddi dysfunction. Inflammatory diseases, those ones which with itis, inflammation in Latin, right? So cholecystitis. Cholecystitis is inflammation of the gallbladder wall. Cholangitis, that is inflammation of the walls of intra or extrahepatic, or both of them, intra and extrahepatic bile ducts. And finally, hepatitis, which is inflammation of the liver structure, the parenchyma. Also, third group is metabolic diseases. Actually, that is a group of diseases which are caused by different disorders in metabolism, which are accompanied by deposition of some usually unmetabolized products in the liver. There may be glycogen, it may be copper, there may be formation of hololids, for example, and so on and so forth. So usually it leads in affection of the liver parenchyma, development of fibrosis, cirrhosis, then decompensation of liver's functions with the development of renal failure, portal hypertension, and if not diagnosed and treated timely, or in case of fulminant progression, they may lead in death. Anatomic and physiological peculiarities of pancreas. Pancreas surface is smooth in early childhood and gets rough up to 10 to 12 years. Parts and participles of pancreas in children are less in number and sizes. There are numerous Langerhans cells. Parenchyma mass intensively enlarges after 4 years. Fibrotic capsule covering pancreas surface is less dense and more elastic in children. Pancreatic duct diameter is not changed along its whole length. More developed endocrine function comparatively with exocrine 1. We may say that actually out of all digestive system organs, the pancreas maturates for the longer period of time in general, and sometimes it's completely formed only after 12 to 20 years of child's life. Here again, if we are discussing syndromes, they are all the same: pain, dyspeptic, and intoxication syndrome. Pain syndrome should be characterized here according to all those parameters which we have mentioned already for all previous systems. Character of pain, epicenter of pain, irradiation, provoking factors, those factors which help to relieve the pain, and so on and so forth. For example, if the child admits that his pain has epicenter at left hypochondrium, it's irradiating to the back, and at the same time it gives a belting pain, which resembles the belt, and that it's accompanied by vomiting with no improvement of the child's condition, high intoxication, and this pain is very remarkable. It's not attack-like, but it's remarkable, high grade, and quite persistent. So you can think already that it may be acute pancreatitis, especially if it's developing in the background of cystic fibrosis, if it is developing on the ground of mumps, for example, because these are basically two reasons which can provoke development of acute pancreatitis in childhood. So that is why you see that on the background of complaints, collecting and anamnesis, you can already guess quite with very high probability that you are right with the diagnosis. Dyspeptic syndrome. Here we may have nausea, vomiting, belching, diarrhea, or unstable intestinal discharges. If we're talking about diarrhea, usually it's a sign, very often sign of exocrine insufficiency of pancreas, and it's combined with very significant, remarkable body weight loss and specific changes in feces. So that's why also characteristics of diarrhea and characteristics of all another objective signs may also help you to guess a diagnosis. And intoxication syndrome I will not stop at because it contains all the same non-specific symptoms which are usually typical for any intoxication of any origin. If to discuss laboratory investigations, they include clinical blood test, and here for pancreas it will be of no specificity at all. Pancreatic enzymes evaluation in blood and urine. The golden standard of exocrine pancreatic insufficiency, fecal elastase 1 evaluation in fetus, lipidogram, coprological or again biochemical stool analysis, which may detect the presence of steatorrhea, creatorrhea, and amyloria. Sugar level in blood fasting evaluation, glucose tolerant test. Last two methods are needed to assess incretoric function of the pancreas. And instrumental investigations, which include ultrasound investigation, endoscopic retrograde cholecystocholangiopancreatography, computed tomography, magnetic resonance tomography, and also endoscopic ultrasound of the pancreas, which is more and more widely implemented into everyday practice. Semiotics of pancreas damages. Before telling about that, I would like to admit that actually among all other organs of the digestive system, pancreas is affected in quite rarely, relatively quite rarely. And as I have already noticed, basically these affections are caused by congenital pathology of the structure, anomalies of pancreatic structure, or congenital anomalies which are accompanied by production of too dense, too thick secret, pancreatic secret, for example, cystic fibrosis and alpha-1 antitrypsin deficiency. Or it may be also caused by complication of mumps viral disease of childhood. So that's why if we're talking about anomalies of pancreas, they're usually hyperplasia or hyperplasia, additive pancreas, sometimes congenital cysts. And if we're talking about inflammatory diseases, these are acute and chronic pancreatitis. Here you should be able to understand, to know the symptoms of exocrine insufficiency of the pancreas, which usually associates chronic pancreatitis in children. And you will discuss it thoroughly on your practical classes. On this, I would like to finish our lecture. I guess that everything was clear and informative for you, and I guess that you will use this information while preparing for the class dealing with this topic. I wish you good luck, good day, stay strong, healthy, take care of yourself, and good luck in your study, of course. Goodbye.